So today we're gonna start like very slow, or there are gonna be time between slides for question, especially about the course and so on. So it's gonna be mostly a refresh on cell biology, and of course all the cell mechanism cannot be covered in course one. So I picked some examples that I think is relevant and like may guide you to have a sort of an overview on what like eukaryotic prokaryotic cell does and where it came from and so on. And yeah, like it basically is what I thought was most interesting as examples, and also what I think would lead you to broader knowledge, especially if you do a little bit of research yourself. But I'll be there for any questions. You can even like private message me with whatever, as long as it's topic related, preferably. But yeah, so first slide, thermodynamics and information. Uh, our world is basically about absorbing and converting energy, especially in the eukaryotic world, thus increasing entropy, which is basically a way that like the way we consume energy is increasing the entropy, which is like basically the disorder in the universe. But we do it in a more complex form, as interaction that is in a sense some information that is more complex than the death of a star or like the birth of a star. Like in the cosmic scale, it's all about destroying and creating at like you know a physicist would say it's like a simple event, but we won't go into that. Basically, what I mean that we can observe, draw a conclusion about these phenomenons, whereas a star or a unicellular organism cannot really have this level of awareness. And we are constantly trying to improve our way of optimizing the energy and resources handed to us, or trying to convert like misfortune into opportunities. Let's say, um, as a more contemporary example, like deforestation could lead to an opportunity of planting more trees that would have like a yield, and you could grow like truffles or like expensive trees that, instead of like breaking down the Amazonian biodiversity, we could basically profit out of turning the planet to a more habitable place. So yeah, we strive to have a better coexistence with our universe, and also like optimizing the energy uh that is handled. Yeah. Any question? So yeah, moving on to slide two. So we come from a long line of changes, right? The street diagram shows like the ancient divergence of bacteria, archaea, and eukaryote, where eukaryogenesis um, led to the mice and chimpanzee and us that from 60 million years ago to 10 million years ago, like a lot of things happen, and I won't go in detail in which genes right now that made us who we are. But let's pick one, for example. Like there's a gene called FOX2, even present in mice, and that is regulating the way we communicate. It enables us to have vocal cord that will like promote like more of a pack existence, like have like animals that group together and screams or communicate in the same tune that leads to a more of a flock awareness. Yeah, any questions?
Uh, what is an archaea cell?
Archaea, they are uh, they're almost similar to bacteria, but they have like let's say a cell wall, and like they have mechanism that allows them to survive in way more extreme environments. Like that, some of them are called extremophiles. Um, so yeah, you'll find them in let's say sulfuric volcanoes, or like super deep underground or underwater, where there is like almost no way a bacteria or eukarya would survive there. But they're also quite prominent. Any more questions? So moving on to
Oh, wait.
scientific. Yeah.
Ollie,
Yeah.
did you
Maybe
just
you have a
ask?
better explanation.
No. Ollie, did you ask what an archaea is or what a eukarya is?
Yeah.
Archaea.
An
With an A.
archaea.
Ah, okay.
Yeah.
Because eukarya is like all the animals and
Right.
plants and things that we can see, right?
I misunderstood you.
Yeah, because I haven't really I haven't been diving deep into what an archaea is actually. But like it is a couple of search away from. We just time for it right now. Unless JPL, you have something to add. My explanation, like I have like a very
I just
basic view on.
understood something wrong.
Yeah, I don't think I said something wrong, but
No.
it's like it's
Everything's
very.
fine.
It was a basic explanation. I can actually I can do some research and come back, or maybe next course with like a broader understanding of what an archaea. But for now,
No, I
it's
just understood something wrong.
Archaea are extremophiles, right? JPL, like basically that's the way we understand.
Aren't they just like prokaryotic cells instead of eukaryotic for the most part?
Yeah, exactly. Yeah. So
I don't know.
yeah.
What else?
Slide 3. So yeah, the importance of scientific culture. Like, do you see a meme like that says "best crossover episode ever"? Like, this is basically the mentality me and JPL we share. Like, we love what is scientific rather than what is sensational. Like, the 3 Blue 1 Brown about and Ben Eater crossover where they talk about Hamming code, but especially eukaryogenesis, where we were thinking about yeast being in the middle of it, as in they still have the plasmids, they still have like many of the organism the organelles from the prokaryotic, but they do have like uh the membrane, and like they're from the fungi kingdom, and they're bad at so since we're part of like the natural life product, as I as I like to think of it, um we want to make as many memes and groups and cultural assemblement, and like basically what we're doing here is we're promoting scientific culture, especially in the biology, to have a bigger importance on like and maybe raise awareness on what actually matters, because as you may have noticed, a lot of the focus is not directed to science and biology. Most people don't care, and memes and assemblement can change that. That's basically it. You have any questions or comments? Okay. So yeah, now with the improvisation, slide 5. So it is everywhere. Uh, oh, actually, no. Yeah, slide 4. Sorry. Artificial selection, or human selection, as some would say. No, it is a slide 4. JPL, just next slide. JPL, hello, hello. Anyone hear me?
Yeah, I can hear you.
Yeah, I can hear.
The one with the brassicas on it, JPL.
Can you hear me?
Yeah,
I can hear you now.
Which one?
brassicas. Wild mustard.
Yeah,
This one?
our artificial selection, the cauliflower.
This one, right?
You guys are on the brassica slide.
Yeah.
Okay.
Yeah. Nope.
No, you're way too far. One more. There we go.
Okay.
I was on this one the whole time.
Can't hear Soizek.
Yeah, can you hear me now?
Yep, we can hear
Yeah,
you now.
I can hear you.
Okay, I'm just gonna quickly escape and rejoin. There you
Yes.
go. Yeah. Okay, so artificial selection or human selection has played an important role throughout human history. From selective breeding to atomic gardening, we made modification on many genes that wouldn't have occurred otherwise through natural evolution. Although, since we're part of the natural life product, we can debate whether it's considered as natural evolution. Like we're technically nature in a way, but that is philosophy. We try to avoid too much ambiguous philosophy. So, any questions before we move on? Great. As a little side note, I probably should have included atomic gardening, but I highly recommend you to look it up if you haven't already. Uh, and yeah, that's it. Slide 5. So, improvisation. Um, a model of the origin of eukaryote through serial endosymbiosis. This is basically how bacteria became animal and plant, or aka eukaryotes. We're very different physically and uh, and composition wise than plant, but at the origin we're actually the same cell type. Um, so yeah, empathizing on the fact that transcription and translation became physically separated in eukaryote, where we traded protein synthesis speed for stable translation, which means that bacteria has a very fast way of synthesizing proteins, since they don't have to exit the any wall or membrane of the nucleus. They're all like very packed together, and their DNA is yeah. You we're gonna come to it later. But the proposed ancestor of mitochondria were aerobic, heterotrophic prokaryotes. I will link in the plasmic cloud like how you define those. But which means they use oxygen to metabolize organic molecules obtained from their organism. And the proposed ancestor of plastids were photosynthetic, photoautotrophic, or photoheterotrophic prokaryotes. Those are big words, but you don't have to learn them by heart. It's just if you come across them, it's just gonna it's just gonna uh, light up a bulb, and you're just gonna know right away what it is without having to learn it by heart, because like, oh yeah, I heard of that. So yeah, it's good words to just have somewhere in your memory, and yeah, it's words are useful, nomenclature is useful, and that's it. And any thoughts, question on anything?
Yeah, what do those words actually mean at all?
Okay. Uh, you want me to explain all the words, or do you have any words in particular that you to have described?
Heterotrophic and the ones like that.
Okay. Okay. It's basically does it well basically does it use light at all, right? So it's like hetero or photo for like does it use light, and does it convert uh like does it convert carbon, like does it use well yeah, it starts with does it convert carbon or not, and then does it use light or not, and then uh, do they yeah, I'll come back to that later, but like I'll link a Wikipedia
Yeah,
article on how to define those.
I don't know if y'all know, I've been writing some stuff and posting some stuff in the voice and video chat thing.
Yeah.
So yeah, I know like in some of the earlier textbooks or whatever, it's like consumers and producers. You have the producers, which it's mainly photosynthetic, but I mean if you want to get really technical, you could have like chemosynthetic, or I think there's like the radioactive fungus thing, but
Yeah. Cool. Yeah. Okay.
Yeah,
So
and
you have actually a
the
product.
the giant top of it is the sun.
Yeah.
So,
Exactly.
Yeah.
Yeah. And then does it fix carbon or not? And then there is a last thing I can't remember. I don't know my word. I used to freaking know those by heart as well. But yeah. Anyways, any more questions?
Can you hear me?
Yeah.
Hello? Oh yeah, I think a
Oh.
basic glossary would help out a lot for some people.
I'm on it. I said
Yeah,
that
yeah.
I will link it in the
Okay.
Thank you.
So as the mandatory shit talker, there is no basic glossary because we're talking about just a tremendous amount of things. This basic glossary is basically going to be 40 pages long. This is just going to be this is one of those things. Bast is taking bioengineering right now. She's trying to memorize all of the amino acids. I'm like, you don't have to memorize all the amino acids. There's just going to be a chart on the wall of the lab that you work in. And because memorizing this stuff makes it easier, like it's going to get ingrained and you're going to understand it. But there's literally, like, we're talking tens of thousands of terms.
Yeah, I'm
Depending on even just like the metabolic pathways of mammalian cells. Memorizing this stuff is insane. There is no. So Google is your friend, and when you see something that you don't understand, put a rubber ducky in there.
yeah.
I mean, just be
short
like, this is the.
of learning Latin, but I mean for the
Also, a really good idea, by the way. Everybody should take Latin and Greek. Yeah.
But
strongly suggest
for the 3, 4, 5 syllable terms just covered in the course, it would be good to have a little just like shortened version because I'm not afraid of those words, but I don't necessarily know what all of them mean, and I have a fair amount of Latin background, but it still bites me in the butt.
A glossary will come up shortly in the channel. I will make sure of that.
If there's enough people that actually show interest, if you guys want to take a very small version of Latin and Greek for biology majors, I will totally teach a Latin and Greek for biology majors thing. Because
cool, I'm down with that.
Because one of the things that I like to be a dick about is when somebody's like, I don't know what this thing means. And it's like, well, it's fucking Latin. Duh. So it's actually very, very useful. And it's not just useful for understanding biology, but it's also useful for understanding language in general, which makes it useful for learning other languages or understanding other languages that you don't actually know, which is very useful for us Americans. And also being able to read scientific papers when you're like, okay, I can just Latin this out, and okay, it's cool. I got this.
I had Latin and Greek in third grade primary school, but it's a long time ago, so I'd definitely be down for a little.
Also, we could just do like a bio-specific, but that's off topic. And so somebody just throw that into, I don't know, the biology channel, I guess. If you're interested, throw something out there, and I'm going to let you get back to it. Sorry for interjecting.
Cool.
Yeah, and also Wikipedia is your friend. Like, it has the definition, it has the Greek root or whatever. So yeah, I'm going to end it on there, not get off topic.
Oh, cool. Thank you. So, any more things before we move on? Okay. So, slide 6, animal cells. So this is basically, in short, what we're made of. You have a nucleus, which is surrounded by a nuclear envelope. The double membrane enclosing the nucleus is perforated by pores and surrounded with endoplasmatic reticulum. So the nucleus is a structure involved in production of okay, I made a little error in the text thing, but yeah, it is surrounded by ribosome that's gonna grab onto RNA and so on. We're gonna come in, like we're gonna go way more in specifics in the next courses. But so you have chromatin, a material consisting of DNA and proteins. It is visible as individual chromosome in a dividing cell. So whenever it divides, you can actually see the chromosomes that you used to see in many papers where you have even the X and Y. So the ER, endoplasmatic reticulum, is a network of membranous sacs and tubes active in the membrane synthesis and other synthetic and metabolic processes. It has a rough ribosome-studded and smooth region, so smooth ER and rough ER. Sometimes cells will have flagellum, locomotion organelle that is present in composed of a cluster of microtubules with an extension of the plasma, within an extension of the plasma. Centrosomes, those are like, well, the cell microtubule are initiated and contains a pair of centrioles, which is like you see it in the graph, right? It's like it looks like two magnets, and I propose something in the systems and complexity that it has some weird quantum effects that it finds basically where is the best region to separate and so on. But anyways, more into this in the private message or whatever. So the cytoskeleton, it reinforces the cell shape, function, and cell movement, and the components are made of proteins, microfilaments, intermediate filaments, and microtubules proteins. You have the microvilli, which are projections that increase the cell's surface area, peroxiomes, which are various specialized metabolic functions, mitochondrions, which are that is where like the cellular respiration occurs. It is basically the industrial production of energy. That's where ATP, adenosine triphosphate, is generated. You have lysosome, which are like near the Golgi apparatus. They're digestive organelles. Those are the munch. They basically break down macromolecules, or another word for it is hydrolyze. They hydrolyze macromolecules and turn them into raw components. The Golgi apparatus is an organelle active in the synthesis, modification, and sorting of the secretion of cell products. And what's in animal cell but not in plant cell, you have the lysosomes. Why did I even write this? Okay, they're a different type of lysosome, but I didn't specify it, so I'm not gonna go too much into how much the lysosome in plant cell differs from the ours. But sometimes they're not even present in plant or animal cells. It's complicated. The centrum, like the centrioles, which are we're gonna go in that later, and the flagella. Sometimes they're present in some plant sperm, but that's it. Like lysosomes are not present in plant cells. Centrioles as well, they're not present in plant cell, and flagella are only present in some plant sperm. My brain just farted. My bad.
Okay, if I may,
I am about to ask for questions. Any questions? You may, Gabriel.
Somebody has a question, I'm gonna listen.
Okay. But
otherwise,
anytime you want, Gabriel.
Yeah, I know. So I got in a couple minutes late, so I don't know if we did this, and we did this the last time we did like plasmid design. But are people just kind of chilling, being like, "Cool, there's a bunch of stuff here," or are we getting like, are we rehashing basic bio, or how's everyone feeling here? This is actually the question, because
we're
a couple of slides in, and some feedback. Okay,
Gabriel,
I think would be.
Gabriel, I did start with saying that we're gonna start very slow, basically learning how how the audience is, like questions about the course, about the slides, and all those questions are accepted. Like for now, we're taking it very slow for course one, because what's coming, it's gonna be progressively
Yeah.
more technical, and
No, that's great. So this is why we need to know how people are.
yeah.
But this means that y'all's need to feedback a little bit. And I know a couple of people in this voice chat have actual like university biological education, but a couple of people probably don't. And so, how's it going, people?
Yeah,
Yeah, I guess I could start like the check off, and everyone could work down from there. I guess that might work. But I did all the way up to like 1 year beginning bio at University UNF, and then that's where I ended. So that's where I am. So this is where I'm pretty comfy. Once where it starts getting kind of probably episode 2 or 3 of this, that's probably where I'll really need to tune in. Next person.
yeah. Yeah, definitely. Like if you've seen the headlines of the 6 different courses,
Dr.
you know that,
McFish,
yeah.
we're looking for you. How's it going, buddy?
Did you learn something? Is it cool? Is it boring? Is it completely inaccurate?
Dr. McFish's microphone is not working. I'll come up with it later.
Do I just speak into the microphone and you guys can hear me? Or
Yes,
Oh, no,
yes,
no,
it's
no.
winning. Oh yeah, now it works. Yes, great.
That's how it works. Yeah.
Okay. I was saying that it was a little bit overwhelming, but I know most of the stuff because yeah, I don't know.
Cool.
Most of the stuff is not new, but it's a little bit too much information in my opinion.
Okay.
Well,
So
while
yeah.
we've got the recording, you can go back over and over until you actually
Yeah.
am comfortable.
Yeah. I'll come up with it later. Do you have any input so far?
hello. I have some
Hey.
inputs.
Yeah.
Cool.
Okay.
Some dude. Yeah.
Yeah. So so personally, I've taken AP Bio in high school, and also I took like Josiah's little like a biohacker class. And also like I watched a bunch of like videos and stuff, like there's a lot of like content on it, and like mess around with like Snapgenes. But I think like where like so I have like some vague concept of how it works, and you can have like bio games in VR where you go inside a cell and you can memorize like all the names, and you can have vague concepts of them just being like, molecules smashing into each other, and they're like, and stuff like that. But I think like where it falls apart for me is like I don't actually like reproduce any of this stuff. Like when I actually do anything biology related in the physical world, like the closest thing is to like grow plants, like water it and put some fertilizer in it, see if it grows fast. So it's like for me, it's like I have like all these abstract concepts for things I can't see that may or may not be how it exists in reality, and and I can't actually like do any of it, let alone do any of it that's like useful personally, or it's not entirely true. Like I learned how to like cut a little branch and put some hormone in it and maybe duplicate it and maybe like give it to friends. So that's like the most useful thing I've done so far. But like
what about eating a sandwich? That's also very useful. Like eating a sandwich, getting sleep, doing push-ups. You're a big bag of cells yourself, so you're doing lots of useful stuff. So.
Right. I mean, the goal of it is like to very gently and progressively end up with an understanding on how would you pinpoint phenotypes, and how to find the genotypes, and becoming a little bit more theoretical about what is genetic engineering, and how would we go about understanding how to make a plant better, or a yeast cell better, or maybe an animal cell better, depending on your
Different.
location and legalities and so on. But how to understand these concepts is basically what this crash course is about, and also having like a feeling of conviviality. Like we're together in this. We're here to bounce ideas off and have a great time hanging out and so on. Like else you would be watching a YouTube video about it, right? So yeah.
Oh, okay. I see. I see.
Yeah. Anyone else got some input? Jasmine, Sai. How's it going, people?
This was my 11th grade junior year high school bio class for the most part.
Likewise.
So
they do repeat that in the
Yeah, but for me that was 30 some years ago. So this is still helpful. I'm not complaining about anything. My only actual question is the T-shaped pasta structure towards the front center.
Yeah.
What is that? That's the centrosome.
Yeah, but that is still quite unclear even for today's very modern and very like how those centrosome works is kind of still some spooky. Yeah,
actually, as a person who has a degree in molecular biology, I could give you a 2-hour lecture on the centrosome, but we're not going to get off topic. I'm just trying to make sure that everybody's on the same page right now. But if you guys want to sit and talk about the centrosome, holy crap, can I?
There's a lot
Can we
of
have
things
something for that another time? Oh, that'd be great. No,
we're just like centrosome 101. It's just gonna
I'd be
2 hours of.
Oh my god. No, okay, so I'll come up with it later. They can't talk because they're in a crowded room, so they're in voice chat, but I'll be like the human voice to text for them. So they were wondering how enzymes work, and they supposedly did AP Bio, so are like somewhat familiar. Mm-hmm. And also just kind of read their voice and video chat, maybe. But
I'd be happy to explain about how enzymes work right now. But the thing is, we're keeping those for.
We got to step through.
So that's for later. Okay, I think that covers them though. Thank you.
Cool.
I'll come up with it.
Thank you, everyone, for taking this little break, so we could just make sure that everyone was on the same.
Oh yeah, definitely.
In the same place. Yeah.
But would I be wrong to say that centrosomes are basically what pulls chromosomes apart? JPL?
It's way more complex than that. It's the stretchy tensile dynamics of the of the cell structure. So very weird.
Part of mitosis, correct?
Say what?
Mitosis?
Yeah. Well, and also actually holding the cell together, and part of why it does or doesn't deal well with cold stress and osmotic stress and a bunch of other stuff.
Oh.
Yeah.
And part of senescence. So yeah, it's like the only reason that we say that we don't understand it is because it's so hugely important. We're in a position where we're like, wow, look at it do 40 things, and it probably does 120 more.
Yeah.
Okay, so moving on.
Yeah. Slide 6. JPL?
Neil, did you die?
Yep. I think. Can someone message him?
Yeah, no, we just pinged him in the chat. We're good to go.
Okay.
I think.
Did he switch? Okay. Yeah, he switched.
Yeah, there we go.
But now I may not. Oh, okay.
Yeah, phospholipid bilayers. Keep going.
Yeah. Yep. So, where were we? Okay, the structure of a phospholipid and the self-assembly of a cell wall. A phospholipid has a polar hydrophilic head and two nonpolar hydrophobic tails. This is the main fabric of an animal eukaryotic cell membrane, and the hydrophobic tails gives the ability to self-assemble while being the flexible and modular way of sticking to each other and avoiding water, and other hydrophobic molecules similar to oil doesn't dissolve in water yet aggregates. So this phospholipid symbol is good to keep in mind as
Why do they self-assemble?
It's if you have a lots of magnets, let's say, but it's more complicated than magnets, but they will all clump together in the plus and minus region, right? And the head is the one that would rather be towards water, but the one that matters the most is the tail in the self-assembly process, because they're gonna they're always gonna make this surface tension circular and kind of fold itself on itself, basically making a little bubble.
Could you explain that again? I don't quite understand how the bottom part, why it makes the tail that way.
Yeah. So yeah, those tails are basically like the way the space filling model shows it. It's like it has a little, or the other one, it has a little angle, right? And the angle is basically that's the thing that's gonna determine kind of the way it bends when they all clump up like magnets, and that's also what's gonna cause the imagine if it's only fat, they're basically gonna form a scaffold automatically while just being close to each other.
May I?
Yeah.
Okay. So we have two parts to this molecule. The top part is the hydrophilic head. The hydrophilic head is, well, it's the phospho part of the phospholipid bilayer. And it's hydrophilic. Now, we're made out of water. And so hydrophilic things like water. Like they're water soluble. They like to attach to water. They like to bump into water. Hydrophilic. Again, going back to Greek and Latin. It water-loving molecule. It's got a water-loving bit on the top. Now, on the opposite end of this molecule, we have a hydrophobic tail. And so this is just a long chain of carbons. That's what that zigzag is. Every zig in that zag is a carbon. And so these are long carbon chains. And the long carbon chains are hydrophobic. The reason that they're hydrophobic, as in phobia, as in don't like hydrogen or water, is that they are non-polar. And you can tell that they're non-polar because they're basically lines as opposed to this complex bendy bit. And so if you look at the more complex model that looks like kind of, I don't know, somebody who's really, really high drawing a picture of Cardi B. This top part has a certain type of shape that attracts water. And the bottom part has a very linear shape that is adverse to water. And so what happens is when they're in a water system, like us, for instance, we're a water system, they bend and they create, and so now you're looking at this third graphic, right, where the things that like not water, those are the long yellow pieces, they like each other. And so they make friends. And the things that do like water, they form a defensive shell on both sides, because I am a I am liquid both inside my cell and outside of my cell. And so it's created, and this is just through pure evolution. It has been a random happenstance of creating a system where it makes bubbles. And we're just a bunch of bubbles. And that's how that works. And it just has to do with these long zigzags being not water friends, and then this complex little area in the gray being friendly with water. And in the same way that you can take a cup of water and drop pepper in it, and then watch how surface tension works, that's how this is arranged.
Yeah.
Cool. Sorry. Back to you.
Okay, thank
good.
you.
Yeah. Like the little last thing is that well, you can see like a straight fatty acid, which is a saturated one, and the one that is like zigzaggy is the non-saturated one, and it, basically, yeah, as Gabriel said, it's the one that mostly will like each other and would rather stay away from water. But that's basically it. Yeah. Yeah.
There's a question from Il Comp Up. I'll come up with it later. He wants to know: is it hydrophobic and hydrophilic as a result of the shape or the charge of the sections of the molecule?
It is the charge.
These two concepts are not able to be separated. The shape, like if you get really good at it, you can tell that the shape of the molecule, how it affects the charge of the molecule. That's what I'm saying. Like these long carbon chains, those are obviously hydrophobic. Like,
yeah,
it has to do with the shape, but the shape defines the charge. So having these two concepts separate actually doesn't help. You need to understand that the shape of things actually affects the way that they work. And so I think a long time ago I talked about like material science and how the shape of objects changes their material properties. It goes all the way down to the chemical level. So once you get enough, and you will eventually, you can look at the shape and you're like, yeah, that's obviously hydrophobic. And it's because the shape makes a charge. It's structural chemistry all the way down.
Okay, cool.
Any more questions?
I have a question.
Yeah.
So for the fatty acids, does it? I've always wondered, is it like the fatty acids are always the same, or does the cell use whatever fatty acids it has? Hello?
So is that?
Yeah, Aniki, you there? Nope, there he is. He's back.
Yo, I noticed. Yeah. So the question was, if I think I got it all. It was that. Well, actually, can you repeat the last bit, just to make sure I got it all?
Yeah. So, what is it? The fatty acids, are they all the same in every cell, or can they differ? Like how many carbons and how many?
Yeah, exactly.
Double bonds in there.
I got it right then. Well, they do differ, and you may have heard that non-saturated fat is better than saturated fat, for example. It's basically because of that you would like to have a little twist on your fatty acid, like the leg is being a bit bended, right? When it's straight, it doesn't allow for a lot of mobility. When it's a little bended, when you have at least one fatty acid that is a bit twisted, it allows the cell to be more flexible. On top of that, you have something we don't talk about right now. We will later on because, well, it's cholesterol. Cholesterol is a precursor to hormones and so on. So we'll attack that later. But what it does, it also secure a more like it secures some integrity in the shape. But more on that later on. The major takeaway
simple takeaway is that there's a lot of different ways to have fatty acids, and sometimes a little bit of kink is good.
Right.
Moving on.
But you a little less thing though. We are able to convert saturated into non-saturated and back and forth when needed. For example, in winter, it is good to have a little bit more saturated fat, as well as they say in the book. But anyways, yeah, yeah. Any more questions before we move on? Okay.
Hello. Yeah, I just have one quick question. So I understand like the DNA holds the information to make proteins, but like but this thing has like fat. So how does it actually go from like the code into like protein into like the structure that has some parts that's not protein?
Well, we have transport proteins that's gonna basically take in all the thing it needs and spit out all the thing it doesn't need. We're also gonna cover that like later on, but yeah, that's basically how we're gonna attack proteins a bit later on. But for
yeah,
now,
I'm gonna make a hard push that we just start moving through.
Yeah,
we've cut a little hiccup where people are like, "Hey, this is the wall, but what about doors, windows, and the roof and the floor make dinner plates?"
Exactly,
so let's move on.
Let's do it, and that is exactly why we haven't started with something more hardcore than this, because we really want to have a feeling on where to go and so on. So yeah, it's perfect. I really appreciate all the input. Can we have a slide 9? Next slide, JPL. Yeah, apparently he doesn't hear us. JPL? He told me he could hear us. Okay, perfect. Okay. So the mitochondria. This is the site of cellular respiration, as they say, but it's basically the energy production center, like the main energy production center on the cascade that we're also going to cover later on. But it has like inner and outer membranes of the structure that has been drawn like at the right with a like tunnel electron microscope. We use fancy terms for basically finding out what's going on in there. The cristae there is a the cristae are enfoldings of the inner membrane to increase the surface area and maximize the amount of reaction that can take place. That means that basically when you have more wrinkles, you have more more space. It's like the lungs. You have more space for a chemical reaction to occur. The two compartments are bound by the intermembrane space and the mitochondrial matrix. Many respiratory enzymes are found in the inner membrane and matrix. The free ribosome are also present in that matrix, where DNA molecule, usually being circular, are attached to the inner mitochondrial membrane. So it has like bits of information from the nucleus that tells them how they should replicate, and yeah, there is also a whole other mechanism for telling whenever the mitochondrion is like failing or too old or just needs to be recycled, and a new one is being spit out. But yeah, basically a lot of surface area allowing a lot of reaction to occur. Major part of the energy called, in the biological world, energy is usually called ATP. Yeah, and yeah, that's basically it. Any questions? Okay, moving on to slide 8. There must be a delay. JPL? I'm looking forward to get some internet. Okay, there you go. Chaperonin, protein that fold proteins, or that actually facilitate the folding of proteins. This complex has an interior space that provides a shelter for a folding of newly made polypeptides. Polypeptides, they're basically long chains that come from the nucleus in the raw shape of a protein, which is like not a shape, basically like a straight line that is ready to bend and twist, but due to like acidity and other chemical factors, it cannot just get the shape it's programmed to get, and the chaperonin facilitate this process in like giving it the exact environment for it to fold without disturbance from the outside activity. So it has like a cap and a hollow cylinder sub domains of the protein, and yeah, the cap opens, the polypeptide comes in and gets folded correctly and gets spits up. Any questions on that or thoughts?
How does the polypeptide get in, or is it from the transport proteins that you mentioned earlier? Like, how does it get the polypeptide in and not anything else?
It is a transfer protein that leads it in there, I would assume.
So it's a physical thing.
So on again, chemistry is a physical act, even when we start to think about it as being just like a weird physics type of math thing. But so this cartoon is not really appropriately expressing how this stuff works. And so there's a whole cascading series. So when you look at a graphic number, I'm going to call it graphic 0.
Yep.
Because it doesn't have a number for some reason. Graphic 0 is actually a very complex structure. Although those little nubblies are pieces of biological material crumpled up into lovely little confetti balls, and then in the same way, so you're watching this 1, 2, 3 of how this works, and this is basically a water bottle, right? It is. I mean, it's a water bottle. You take the cap off the water bottle, and then the water goes into the bottle, and you put the lime in the coconut. And so what this is actually talking about is how there is a structure, and things open the structure, and there's a whole, we're talking a whole semester of college about how this structure opens, and you just you're gonna have to roll with it, man, because it's not actually important. You just need to know that it exists. Right. Something takes the lid off the bottle. The thing goes into the bottle. That's number 2. The lid goes back on. When the lid goes back on, it makes the shape. And I know that sounds really weird when I say it this way, but that's what's happening. It creates a conformational change, which causes your polypeptide to be pushed into a certain shape. And then the lid comes off again, and off goes your correctly folded protein.
So basically, yeah.
Yeah.
I didn't mean
yeah, so this is just like it's a very simple system. We could literally spend like 3 weeks talking just about how this works.
And the book that I choose for the most of this those slides are it's as it's repeated 8th edition by Campbell and Reese, and it's designed for cell biology. It has 1,500 pages, and it doesn't. It's not covered. It's not covered like how these chaperonin actually like works. It's not covered in this book. It's only like mentioned as
because there's a whole nother textbook that just talks about this. So
yeah.
So this gets into the region of some of this shit is witchcraft. And some of this shit is you're gonna have to read like 40 textbooks, and it's just not gonna be handled here.
Yep.
The chaperone, yeah, but you should read all of the textbooks.
Oh, yeah.
But how it really works is there is a bottle, it makes the shape. It's a magic bottle that makes the shape, and that's it.
Yeah.
That's all you need to know.
We'll get more into the folding, how it folds, and so on, but those are for a later lecture. So it was basically to give the
because my understanding, he's just trying to give you some background here. My understanding is we're trying to get back to protein synthesis and building plasmids and stuff like this. And so this is just a baseline. But if you guys want to learn how to make bottles, magic bottles that make proteins, that's a whole nother.
But it's definitely also why it is definitely why we're looking at it right now. It's also to encourage.
Right. You just have to know that they exist. They're there. It's a thing.
And if you're inspired, like if you find something about it that you feel is interesting, you're always welcome to message me with whatever you find. It amazes me.
Likewise, if someone's like, "Hey, I really want to make magic bottles,"
yeah. So
I'm like bonking my head on the floor. But if you guys, if somebody really wants to learn how to make magic bottles, we're gonna start with Latin, and then we're gonna work our way through the witchcraft. But if you want to learn how to make magic bottles, I can help you learn how to make magic bottles. But we're gonna get there. Awesome. But right now, we're just trying to learn the general flow of things. You know, this is kind of a, hey, you're telling me how to fill up a water bottle. How does glass made? It's like, no. Chill. Just be chill.
Yeah. So, any questions on the eukaryotic world before we move to the other eukaryotic world of plant cells?
I'm sorry, just a quick question. So for this magic bottle, are there various versions of it for different types of like a polypeptide, or is it just like?
Oh yeah, no. There's as far as you're concerned, there's an infinite number of magic bottles because every shape requires either a different bottle, or sometimes it's the same bottle with a different lid, or maybe maybe your hand taking the lid off of the bottle. So this is talking about, this is an analogy for enzymes, changes the way the bottle opens. It's infinite bottles. There's a lot of this.
Right.
This is why I'm saying there's an entire textbook about this thing.
Yeah, I bet.
And I was trying to make it a good analogy. And let's go.
Yeah. I'm not sure if this is too complicated, but why does the polypeptide not fold right away after it's synthesized?
That's quite that I can try to explain it. It's basically because the acidity or basic environment, like it doesn't have the exact environment to get the exact shape that is, like we're gonna go more into this. Like basically later on, we're going to describe the four part of protein shaping and assembling. But this
future question.
You need the right environment to fold it in the right way.
Yeah. Here, let me put it this way. You know how people are like, oh hey shit, I should drink some alkaline water because that's gonna make me feel so good. Like, look, no, your body is at 7.4.
No, don't blood. Even. Okay, no, I work at Walmart, so some of the stocking, like all this bullshit pseudoscience.
Okay, yeah, no, right. So your body is at 7.4. You don't actually Like when you put the water, whatever, when you drink anything, it doesn't affect your alkalinity. However, so this is how the woo happens. However, alkalinity within certain structures, hey, magic bottles, actually changes because they need to change the chemical structure due to pH. But it's only happening within one small structure. It's not happening inside of your body. And so we have this whole series of systems that change and shift molecular structures based off of pH. But they're all in tiny, tiny little bottles. They are not just randomly in your body.
Not in 1 liter bottles that I have to handle like 50 of each day because some Karen wants to get their
no, dude, no. Like if you're not buying pH 9 water to balance your system, like, are you even living?
You're meaning to tell me my imported via bunker fuel filled cargo ships artisanal raw hydrogen infused low pH water is not going to magically cure me of everything?
We're gonna. I don't know. Should we hold for like Ollie? What's the timing? Are we reached 1 hour, haven't we? Ollie?
Yeah, it's 1 hour, but I can keep going.
Okay. How you guys feel? Like, should we hold for today and get the plant cell world next time, or we good to just proceed? I mean, it's up to you guys. I was planning on 1 hour, but it's up to you. What's the feeling?
I'm good to go.
All right, let's continue for a little bit then. But if anyone thinks, okay, it was enough for today, please say so we can, like, at least, not overburden. Yeah, okay. So moving on, JPL, next slide. He really doesn't hear us, though.
No, I'm pinging him each time to change the slide.
Okay,
thank
you.
Did, and he's not changed it.
Mm. I'm gonna try to talk him.
What would it be possible to broadcast the same slide on someone else's screen, or?
Yeah.
Yeah, I mean, Aniki, why don't you just pull up some slides, livecast? Let's do this.
He's on a phone.
Yeah.
Oh, he's on a phone.
Yeah, that is my problem right now. I don't have internet.
Getting on it.
So I think we should hold for today and proceed next week with the last bit, and then perhaps course 2.